Assalamu alaikum. So we try to continue the fourth lectures, and this time we'll talk about tricarboxylic acid cycle. Our reference is Lippincott illustrated review. So I just start with the question: what enzyme convert pyruvate to acetyl CoA? Is it pyruvate dehydrogenase, catalase, pyruvate kinase, pyruvate carboxylase, or aldolase? This question from the previous lectures of glycolysis. Because we define that glycolysis, the conversion of glucose to the pyruvate. And pyruvate, they will go to the acetyl CoA. So just to try to memorize which is the correct, A or B or C or D or E. Take your times. Yes, so let me see, sir. Pay to the pyruvate. As we told you in the last lectures, pyruvates, they can enter citric acid cycle after conversion into acetyl CoA by pyruvate dehydrogenase. Number 2, conversion to fatty acid ketone bodies or cholesterol. Because they go to the acetyl CoA, they can form fatty acid ketone bodies. Conversion to lactate by lactate dehydrogenase. Conversion to ethanol in yeast and some organisms. That's the fate of pyruvate. Fate معناته مستقبل. What is the else? What's else for the pyruvate after formation from glycolysis? Regarding citric acid cycle or tricarboxylic acid cycle or Keb cycle, all of these named by this man, Hans Krebs. This man also this scientist also discovered the urea cycle. So there is a link between the citric acid cycle and the urea cycle. It is a cycle. So let us see overview. Tricarboxylic acid, TS, Keb cycle, citric acid cycle, it's the same name. It plays several roles in metabolism. It's very important. It is a junction. It is a cycle. So it's very important in the metabolism. It's found in the mitochondria. It's the final pathway where the oxidative metabolism of carbohydrate, amino acid, fatty acid converts their carbon skeletal being converted to CO2. So it's the final pathway for the metabolism of macromolecule. If it is carbohydrate or fats or some protein, the carbon skeletal, they produce energy and release CO2, which is exhaled outside. So they need oxygen for respiratory chain. They need oxygen. This oxidation provides energy for the production of the majority of the ATP in most animals, including human. Tca cycle is aerobic. That means they need oxygen for final acceptor, for electron acceptor. In eukaryotic, citric acid cycle occurs in the matrix of the mitochondria. Where it is occur? It is in the matrix of the mitochondria. Keb cycle occurs in the matrix, and they need the final oxygen as the receptors. So look at this. Look at these connections. When you eat foods, for example, protein or carbohydrate or fatty acid, all of these, they go to the citric acid cycle. From citric acid cycle, they produce energy through the electron transport chain that produce ATP. Why we need ATP? We need ATP to take the vital energy. We call it vital energy. We call it vital work for the body, for the movement, respiration, thinking, muscle contraction, heart pumping. So energy is very important for living organisms. Without energy, we cannot live. So that's the slides. They show how it's going glycolysis. We know that glucose converted by through the glycolysis to the pyruvic acid to the acetyl CoA and citric acid cycle. That is the citric acid cycle. You take it from the pyruvate convert acetyl CoA. Acetyl CoA enters the citric acid cycle through to converting to the citrate. It's oxaloacetate and citrate. After that citrate, they give you different 6 carbons. Sorry, it is a 6 carbons. Here we remove CO2 and become alpha ketoglutarate. It's a 5 carbons. Sorry, alpha ketoglutarate. It's a 5 carbon succinyl CoA. So how many carbon? How many carbon released? Just look at it. How many carbon dioxide released? It's a 2. That means acetyl CoA is a 2 carbon. Oxaloacetate is a 4 carbon. It become 6 carbon, and this is called citrate. Citrate, 1 carbon release, you give you alpha ketoglutarate. Another 1 carbon release, they give you succinyl CoA, which is a 4 carbons. Fumarate malate is a 4 carbon, and oxaloacetate. Yeah, these are the reactions. So you have to look at enzymes to look at how many water produced from Keb cycle, how many carbon dioxide is produced, how many energy is produced through the through the Keb cycle. Look at it. It is energy produced when you look at the isocitrate to the alpha ketoglutarate, NAD produced. Alpha ketoglutarate to NAD, another NAD produced. Succinyl CoA to succinate, GTB produced. Succinate to fumarate, FAD produced. We know that NADH produced 3 ATP. FADH produced 2 ATP. GTB, this substrate level, give only 1 ATP. Only 1 ATP. So the citric acid cycle supply intermediate for a number of important synthetic reactions. For example, the cycle function in the formation of glucose from carbon skeleton of some amino acid. We call it glycogenic amino acid. It's a building block for some amino acid and hemoglobin, heme part, which is found in red blood cells. So let us see energy production. If you calculate it, as I told you, how many NAD, 3 NADH is produced, 1 FADH is produced, and another GTB is produced. So the sum of the energy produced from Keb cycle is 12 ATP. You can go and check it. Let us talk about the regulation of the Tca cycle. Mainly several enzymes control the Tca cycle. The most important of these enzymes are citrate synthetase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase complex. That 3 enzyme is very important for regulation of Tca. That's 3 enzymes. You should remember this. Which enzyme regulates the Tca cycle? Important enzymes. It's citrate synthetase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase complex. Citrate synthase is inhibited by ATP. More ATP, that means they stop the cycle. Isocitrate is activated by ADP. When we need energy, they activate it. Also inhibited by ATP. The third important enzyme, alpha ketoglutarate dehydrogenase, is inhibited by succinyl CoA and ADH. So the availability of adenosine diphosphate is very important for Tca cycle to proceed. Inhibitor of the TCA. How can we stop if we didn't need energy? Actinase is inhibited by fluoroacetate. That is the compound. This compound inhibits the TCA. So they can go no respiration. So they will go to the death of the cells. They stop the energy. Actinase is inhibited by fluoroacetate. This is a non-competitive inhibition. Alpha ketoglutarate is inhibited by arsenate. Arsenate is a metal. It's a toxic metal, so they can inhibit Tca cycle. Succinate dehydrogenase is inhibited by malonate. This is a competitive inhibition, so they can be overcome. Inshallah, when we go to the enzyme, we talk about the competitive and non-competitive inhibition for reversible or irreversible for enzyme. What is the significance of the Tca cycle? Complete oxidation of acetyl CoA, ATP generation, final common oxidative pathway to go for respiration chain, integration of the major metabolic pathway. Fat is burned on the wick of the carbohydrate, so we can produce much more fat. Excess carbohydrates are converted to the neutral fat. No net synthesis of carbohydrate from fat. You cannot synthesize carbohydrate from fat, but we can synthesize fat from carbohydrate. Carbon skeleton of amino acid finally enters the Tca cycle. As I told you, this we call the glycogen. So this is a short lecture regarding the Tca cycle. That's what about the, after the Tca cycle? Tca produce that compound, that electrons that will go to the electron transport chain for respiratory chain that produce finally energy ATP. That is energy produced from 1 mole of glucose that break down to the 1 mole of glucose that break down to the produce CO2 and H2O. That 1 mole of glucose completely break down to the CO2, H2O and energy. What is the energy produced? ATP glycolysis will give 8. Pyruvate to acetyl-CoA will give 6 through the NAD. Tca cycle, they will give 12 ATP. That means energy produced from one movement of the cycle of the ATP for the Tca is 12 ATP. That is the total net of breakdown of glucose to CO2 H2O is 38 ATP. Thank you. So until the next lecture.